My name is Laura Newcomb, and I'm a molecular virologist from California State University, San Bernardino, where I have an NIH-funded research lab that studies influenza molecular biology. So the influenza A virus is part of the family Orthomyxoviiridae. One thing about influenza is that it is a pleomorphic virus. So although you typically see it as spherical, in reality, it can actually be both spherical and filamentous, as you see here in this cryo-EM image. Now, in the Baltimore classification, influenza is a class 5 virus. This is a negative sense RNA virus, so it has a single-stranded RNA genome. Now, influenza virus actually has a segmented genome, and so there are 8 RNA genome segments. Due to things like splicing and alternate splicing and translation and alternate translation through alternate translation initiation events or ribosomal frame shifting, those 8 RNA genome segments produce at least 10 mRNAs that encode for at least 12 different proteins during the virus infection. Now, in the virus, in the virion itself, because it's negative sense, that means that the genome must be packaged with its replicase enzyme, an expression enzyme, and that's the RNA-dependent RNA polymerase. And for influenza, that's a trimer made out of 3 proteins, the PA protein, which is standing for polymerase acidic, and then PB1 and PB2, which are polymerase basic. In addition to the RNA-dependent RNA polymerase that's bound to each genome segment, the RNA is also coated with many copies of the viral nucleoprotein, and together that complex is called the viral RNP, ribonucleoprotein complex. And so there are 8 of these in the virion. Again, to get those 10 mRNAs from 8 segments, 2 segments M and NS, each encode 2 proteins. They encode 1 protein from the spliced mRNA, that would be M2 or NS2, and then they encode 1 protein from the intron-containing full-length mRNA, and that would be M1 and NS1. The multiple proteins are obtained 2 segments, PA and PB1, each utilize alternate translation mechanisms to express more than just the PA and PB1 proteins. The other 2 segments are HA and NA, and these encode the viral surface proteins, hemagglutinin, which is important for receptor binding and entry, and then neuraminidase, which is important for exit of the virus budding from the cell. These 2 surface proteins are the proteins that your immune system will produce neutralizing antibodies against. And so these are the proteins that when a vaccine is developed, they target these surface proteins so that you produce neutralizing antibodies. However, these surface proteins are highly variable, and currently there are 18 known HA variants and 11 known NA variants in influenza A viruses. And you may know these segments, HA and NA, if you've heard about influenza being, say, an H3N2 virus or an H1N1. That's referring to what variant of HA and NA that virus contains. And so here is just an example of how influenza viruses are named. First is the type. Today I'm only talking about influenza A virus, but there are influenza B viruses that also infect humans. They have not been responsible for any past pandemics. And then there's also influenza C and D virus, which infect other animals. So we're focusing on influenza A, but if it was a different type, that would be listed here. If it's not human, you'll see what host species the virus does infect and where it was identified, which serial number it is, and then what year it was isolated, along with the H and N variants. And so again, not all of those HA and NA variants are in influenza virus that can infect human, but influenza viruses have many animal hosts. So I have here just some, including humans, ducks, chickens, pigs, horses, dogs, cats, and even seals. Having so many animal hosts does contribute to influenza having rapid evolution. Other characteristics that allow influenza to have such a very rapid evolution and be considered still an emerging threat is that it's a negative sense RNA genome. Having an RNA genome, it has an RNA-dependent RNA polymerase to replicate that genome, and this polymerase has no proofreading activity. And so this does lead to errors more frequently during replication, and this is termed genetic drift, those small errors during replication. Influenza also has a segmented genome, and if there are two different influenzas in the same cell, you can get reassortment of those segments, and that can lead to very dramatic changes that we call genetic shift. And so the natural reservoir for influenza is thought to be wild aquatic birds, and pigs, however, are susceptible to infection with avian influenzas. So it's thought that, evolution in the pig, which is not an avian, it's a mammal, resulted in influenzas that then could infect the human population. Now, the pig still remains susceptible to infection with avian influenzas, swine influenzas, and human influenzas. And so pigs are considered by many to be a mixing vessel for those reassortment events. And that's what occurred with the pandemic in 2009, the novel H1N1, or it was sometimes referred to as the swine flu, because it had segments from avian influenza, human influenza, and swine influenza. So it was considered a triple reassortment. Now, in addition to wild aquatic birds, our poultry friends can be infected with avian influenzas, and we have much closer contact with them. So although it's rare, and I'll tell you about why in a few more slides, but although it's rare, direct avian to human transmission has occurred, typically with not very good outcome for the people infected. And so these are highly pathogenic avian influenzas, HPAI, including H5N1, which was seen earlier, but fortunately recently does not seem to be causing too many issues. However, there is another H7N9 that emerged later that is still of great concern. So currently, these avian influenzas are showing very high mortality for humans that are infected, but fortunately they do not appear to have the ability to transmit very readily among humans. And so only people that were in very close contact with infected poultry became infected. And so, the concern is if a virus with that kind of mortality rate were to obtain characteristics to transmit readily. And so viruses can obtain new characteristics through those two mechanisms I told you about, antigenic drift, where during replication the polymerase makes some errors and that can change the encoded protein, and so now you may have a new surface protein that you don't have neutralizing antibodies against. And similarly, antigenic shift can occur when we have one host cell. You could think here the pig, for example, and being infected with an avian influenza and a human influenza. And so now we get a reassortment event and a brand new virus that we haven't seen before emerges. And so these two molecular mechanisms to produce genetic change can lead to novel influenzas and resulting in influenza viral pandemics. And so this is the 1918 pandemic that you'll watch a video about as well. But this is entirely an avian influenza. So this was, a change in one of the proteins that allowed it to then jump to humans. And the other two pandemics in 57 and 68 were reassortment events between human and avian influenzas. And again, the 2009 H1N1 actually had segments from two different swine influenzas, a human influenza and an avian influenza. And so because of these characteristics of influenza, that it has multiple animal hosts, that it has an RNA genome, so without the RNA-dependent RNA polymerase doesn't have any proofreading, and the genome is segmented, so reassortment happens frequently, all of these characteristics make influenza virus still a concern for emerging. And so here's just another depiction of all the different animals, and it's not an extensive list, but the animals that can be infected, and this gives you an indication of what variants of H surface proteins are found, and Na proteins as well. And so, zoonosis does happen, and influenza is still a concern to have an emerging influenza come from another animal and start to infect humans. And so I just want to stress that this really could happen anywhere because humans live near animals all around the globe. And this is just my example I like to show of me hopefully never being patient one for a jump back to from dog back to human. So let me tell you a little bit about the molecular biology of influenza. And so this is a cartoon depiction of a simplified life cycle for influenza where the surface protein HA binds to the host receptor at attachment. And then we have endocytosis and a fusion event that requires the HA protein and also the M2 ion channel to pump in protons for that fusion event to occur. Once the 8 VRNP segments are released, they're imported into the nucleus of the cell where the viral RNA-dependent RNA polymerase can transcribe and replicate the genome. Transcripts are exported for translation, and the replication products are assembled, and we have assembly of the virus at the plasma membrane, and then budding, which requires the viral neuraminidase protein. And so the HA surface protein is for attachment and fusion, and the HA protein has a receptor binding site, which will bind to sialic acid groups on proteins on the target host cells. And so there's lots of different HA variants, and they can vary at the receptor binding site. And this variation will cause the HA protein to prefer different receptor modifications. And so, for example, the avian HA proteins prefer to bind to the alpha-2,3 modification of sialic acid, which is found in the upper respiratory tract in avians, but is not actually found in the cells of our respiratory tract. Human HA does prefer to bind to the 2,6 modification, which we do have present in our upper respiratory tract. But because we don't have the modification that the avian HA protein can bind more readily to, this explains why the direct avian to human transmission is rare. Because you have to have infection in the lower respiratory tract. So thankfully not readily transmissible. The HA protein also needs to be cleaved by a host protease before we can have the next step, which is fusion. And so human HA proteins typically use a protease that's restricted to the respiratory tract. And this is why in humans, influenza virus replication is restricted to the respiratory tract. However, for the avian HA protein in these H5H7 avian influenza strains, that HA is shown to have a cleavage site that can be cleaved by proteases that are more ubiquitous and found in many different tissues. And so this could explain why when humans are infected with these highly pathogenic influenzas, why they're seeing such high pathogenicity, because the virus can target additional human cells. And so the HA protein of avian influenza can use a very common protease. So that's not very good as far as how many host cells it can attack if it infects a human. But fortunately, since the receptor binding still prefers receptors that are only found in the lower respiratory tract, hopefully we won't see emergence with these highly pathogenic avian influenzas. And so the cleavage event cleaves the HA protein. This is showing you a monomer of what's going to be the HA1 and HA2. And then after cleavage, the M2 ion channel will pump protons into the endosome, and that will alter the structure of the HA2 monomer and initiate fusion. So you get that fusion peptide entering the membrane, and we're going to release those VRMPs. So once the VRMPs are released, they are imported into the nucleus for transcription and replication. And so all of the proteins on the VRMP actually have at least one, if not multiple, nuclear localization signals to target that complex into the nucleus, because transcription and replication of influenza occur in the nucleus. And so the influenza virus genome is negative sense, that's depicted here. And so the first thing that occurs to have viral genome expression is that we have to have transcription occurring to get a messenger RNA to encode the viral proteins. And so the viral polymerase is comprised of PA, PB1, and PB2, and it uses a mechanism called cap snatching, where it steals a 5' methylated capped mRNA fragment from newly synthesized host mRNAs. So if you remember from a molecular biology review, during mRNA synthesis in eukaryotic host cells, the cap modification occurs very early during transcription. And so the influenza polymerase is actually in the nucleus and makes contacts with the host RNA polymerase 2. So it's near these newly capped RNAs. The PB2 subunit can recognize the cap and bind it, and the PA subunit has endonuclease activity to cleave that RNA. And then that small capped RNA fragment is used as a primer for transcription. And that's why all influenza mRNAs actually have a little sequence of host mRNA at the start of their transcript. And so that's indicated here by the little black line. Now the transcription occurs until the polymerase reaches a U stretch on the negative sense template. And here the polymerase stutters and goes back and forth over that sequence to produce a poly A tail. And so if you'll notice the difference between the cRNA replication product and the mRNA messenger RNA product is that this messenger RNA has a cap that's been stolen from host mRNAs, and it has a poly A tail that was generated through repeated stuttering over this U stretch. So it doesn't have these end sequences. Instead, it has a poly A tail. Now those mRNAs, two of them, I told you, M and NS, will undergo splicing in a percentage of the product. They all need to be exported through the nucleus into the cytoplasm for translation. And so it is presumed that influenza mRNA splicing occurs through the standard host splicing pathway. And the nuclear export of influenza mRNAs is not completely clear, though many of them seem to use the typical host nuclear export factor pathway. However, some influenza mRNAs, as I've told you, NS1 and M1, are actually encoded by full-length transcripts that contain introns. So what you're looking at here is the example from HIV mRNA expression, where some HIV transcripts are fully spliced, but others are only partially spliced or not spliced at all and contain introns. Now, these introns actually have elements that are recognized by a HIV accessory protein that can bind to that full-length mRNA and help get it transported out of the nucleus through a different mechanism. So for influenza, there's really very little research that's been done on splicing and export, but we do know that a number of, or that some of the influenza mRNAs do utilize NXF1. And we also know that influenza mRNAs don't utilize that, that CRIM1 pathway that HIV used for mRNA. I will show you that the CRIM1 export pathway will be used to export the VRMPs during viral assembly. So we know that some of these mRNAs do not have an export pathway identified yet. And so, you know, in my opinion, this is an area that could use more research, because elucidating the mechanisms of influenza mRNA nuclear export might lead to the discovery of novel host export factors and maybe even host factors that could be targeted to block influenza. And so after nuclear export, the viral mRNAs are then translated. The mRNAs that have proteins targeted to the plasma membrane are going to be translated by the rough ER, whereas others are translated by loose ribosomes. But what I'm showing you here are the two segments that are known to have alternate translation products. And so there are a couple products that are shown for both PB1 and PA. And just know that so far, PB1F2 has been characterized somewhat and plays a role in cellular apoptosis. And PAX has been characterized somewhat and plays a role in viral pathogenicity. Now, these other alternate translation products, the N40 and the PA-N182 and N155, are not well understood yet. And so many of these alternately translated products are not expressed in the virion. They're considered non-structural proteins that are expressed only during viral infection. And one of the most well-known influenza non-structural proteins is the Influenza Ns1 protein, and that's because it's a multifunctional protein that inhibits the innate immune response at many different levels. And so here I'm just going to show you one activity of the Ns1 protein. Whoops, I can't go back. Well, the Ns1 protein inhibits host polyadenylation, and so that will stop host messages, but won't inhibit influenza polyadenylation, since the influenza polymerase does that itself through the stuttering mechanism. So NS1 is a non-structural influenza protein that is important in countering the host antiviral response. So now that we've had, in this life cycle, we've gotten to expression of the viral proteins, and so now that we have expression of proteins like NP and the RNA-dependent RNA polymerase, there's a switch then from gene expression to replication, where we're going to get an intermediate product that's then going to be used as a template to make more of the viral genome. And both of these steps are initiated without a primer, and so they have 5' triphosphate ends, and they require the nucleoprotein to bind both the cRNA and the vRNA template. The nucleoprotein also makes direct protein-protein interactions with the viral polymerase, and that's thought to promote this unprimed initiation for replication. So after we have replication, and now we have new VRMPs being made and assembled in the nucleus, they need to be exported so that assembly can occur. And so to export the VRMPs out of the nucleus, the viral Ns2 protein, which is also called NEP because of its role as the nuclear export protein, via protein-protein interactions. And so the M1 protein of influenza will interact with the NP protein in the VRMP, and NS2 or NEP interacts with M1. NEP also interacts with that host export factor, CRIM1, and so this leads to export of the VRMPs via this CRIM1 pathway. So after export, the VRMPs need to be assembled into the budding virion, and so in this depiction, you're looking at the HA and the NA, those are the orange ones, surface proteins. Again, M is attracted to the HA and NA, and it is bound to the VRMPs through NP, and so it will bring the VRMPs toward these surface proteins. Now, M2 is recruited at the neck here to promote budding. So M2 has an amphipathic helix, so both hydrophobic and hydrophilic, and will help in making that bud. What you're looking at in this depiction are the filamentous buds. Remember, influenza is pleomorphic, and so it can bud both spherical in shape and what you're seeing here, the filamentous budding. So now to get release, we need the activity of the other viral surface protein, the NA or neuraminidase. And what neuraminidase does is it's going to cleave that receptor so that you don't reinfect the cell that was already infected. And so this releases the virion from binding to the host cell, and you get continued viral replication. Okay, so how do we protect ourselves and our cells from viruses like influenza? And so we have vaccines for influenza that are very effective, and these are given as either a shot, and that's the inactivated virus, so that's a dead vaccine virus, but there's also a live attenuated vaccine for influenza, which is given as a nasal mist. This was taken off the market for a few years, but I believe it's back on now. So really this one is for people that have allergies to eggs, because the inactivated virus vaccine is produced using eggs. And so in both cases, your body will produce antibodies against those HA and NA surface proteins. But as I've told you before, those are highly variable proteins, and your antibodies against one of the variants may not protect you against others. And so as new variants arise, we need new vaccines. That's why every year there's a new vaccine. And, the production time currently is about 6 months for a vaccine that, they know how to make, and they just have to change, which HAs and NAs are being targeted. Of course, there's continued attempts to find a universal vaccine for influenza so that you could just take one vaccine and not have to take it yearly. So those focus on conserved regions of the surface proteins, which again are minimal because they're highly variable proteins. But the stalk of HA is a pretty conserved region, and so that's been identified as a potential vaccine target. And also M2, because it's more conserved. However, scientists have been working for a long time to identify a universal influenza vaccine. So in the meantime, we also have antiviral therapies, and antiviral therapies are typically effective against multiple HA and NA variants because they're inhibiting a viral protein activity. And so they can be very appropriate, particularly for new emerging influenzas that we don't yet have a vaccination for. However, just like antibiotic resistance, the more that antivirals are used, the more they are going to be selecting for viruses that have resistance against those antivirals. And so the current antivirals for influenza are the neuraminidase inhibitors. You might have heard of Relenza or Tamiflu. And these inhibit neuraminidase from cleaving the receptor, and so you don't get the virions released from the target cell, and so that inhibits further viral replication. And so on this simplified life cycle, I want to show you the first class of antivirals. They targeted the M2 ion channel to inhibit that release of the VRMPs. Now, these were the first class of antivirals against influenza, and so they are now totally ineffective as 99% of circulating strains have the resistance against these antivirals. Now, the class that we currently seem to use the most is targeting that NA, and so again, that's Tamiflu is one of those. And so in my lab, we were focused on looking at the activities in the nucleus to look for new targets for new antivirals. Of course, so we look at this VRMP complex. There's a lot of potential targets on the VRMP, so we could inhibit any of these protein-protein interactions that are essential for function. We could inhibit the cap binding ability of PB2 or endonuclease ability of PA. PA also has a reported protease activity, so maybe protease inhibitors would be effective against influenza, although this protease activity is not well defined or characterized as far as what its role is in influenza infection. In my lab, we are focused on NP and some of the targets and activities of NP that could be inhibited. But of course, we're not the only lab interested in looking at targets of the VRMP, and actually one new drug made it to market in 2018 that targets the endonuclease activity of PA, and that's called Xofluza. And so hopefully that antiviral will remain effective for many years to come. And so just kind of to conclude with some key questions, how can we better prepare for an emerging influenza virus? Do you ever think we will get to a universal vaccine for influenza? Will resistance to Xofluza develop? How likely is that to occur? And do we need to continue to identify new antivirals? Lastly, what about the role of surveillance in looking at emerging viruses? Influenza has many hosts, and the next emerging pandemic may not come from the expected host. Maybe it will come from something less expected. And so with that, thank you for listening.